Hello everybody and welcome back. Now we're going to talk about the regulation of the coronary blood flow. The regulation of the coronary blood flow is done mainly by adjusting the resistance of coronary vasculature by relaxing or contracting the vascular smooth muscle in several manners. So there are autoregulation, which have metabolic and myogenic, we have neural control, and we have humoral control. For the autoregulation, we're going to start by the autoregulation. The autoregulation is governed by the fundamental relationship that myocardial oxygen supply rises and falls in response to the myocardial oxygen demands. So it depends on the myocardial oxygen demand. For the autoregulation is the ability to maintain myocardial perfusion at constant levels over a wide range of driving pressure values, which are the mean aorta pressure between 130 and 40 millimeter mercury. For the myogenic control is the atrial smooth muscle reacts to increase intraluminal pressure by contracting, which forms an increase in resistance, which makes the blood flow return toward normal despite the higher perfusion pressure. How does this work? Increase the length of vascular smooth muscle cells activates mechanical gated channels anchored at the cytoskeleton of the vascular wall, which determines an increase of the vascular tone. Now we're going to talk about the metabolic control. The metabolic control is a sum of mechanisms allowing a prompt adaptation of oxygen supply to the energetic needs of the myocardium. The metabolic control is necessary because the myocardium depends almost entirely on aerobic metabolism. It's also necessary because the myocardial oxygen extraction is very high, which reaches to levels of 70 to 80% compared to 25% in systemic circulation, and because oxygen stores in the heart are meager. Metabolic control is mediated by adenosine, nitric oxide, vasodilator prostaglandins, ATP-sensitive potassium channels, myocardial oxygen, and carbon dioxide tensions. For the adenosine is the principal metabolic mediator. It is formed by degradation of adenine nucleotides when ATP consumption exceeds cell capacity to resynthesize high-energy phosphate compounds due to increased heart activity, insufficient coronary blood flow, and fall in myocardial oxygen pressure. This from the myocardial cells, adenosine diffuses into the vascular smooth muscle cells, which causes the vasodilation, which increases the oxygen supply. And now we're going to talk about the neural regulation. The neural regulation has the sympathetic nerve stimulation and the vagus nerve stimulation. For the sympathetic nerve stimulation, on the myocardium it increases the metabolism, which increases the contractility and increases also the heart rate by beta-1 stimulation, and indirectly produces metabolic vasodilation. On the vascular smooth muscle cells, through alpha receptors, activation produces vasoconstriction. Under normal circumstances, the metabolic vasodilation prevails the alpha-mediated vasoconstriction. And now for the vagus nerve stimulation. The vagus nerve stimulation has a mild vasodilatory effect due to the meager vagus nerve distribution into the heart, while acetylcholine administered directly elicits a pronounced vasodilation. And now finally with the humoral regulation. The hemorregulation has the endothelium-derived factors, which are vascular endothelium has a very intense biological activity, performing an array of homeostatic functions in normal blood vessels. Endothelium-derived relaxing factors and endothelium-derived constricting factors are factors derived from the endothelium that can cause relaxation or constriction. For the relaxing factors, they are the nitric oxide, prostaglandins, and endothelium-derived hyperpolarizing factors. And for the constricting factors, they are endothelins. Other factors that can also affect are angiotensin II, which causes vasoconstriction, and bradykinin and serotonin, which cause vasodilation. So this was everything for today. Thank you very much and hope to see you soon again in the next video.